Hi, everybody. I'd like to start by thanking the organizers for giving me the chance to talk to you about our recent work, and also to apologize that I'm not there in person due to some other previous commitments. I'm going to share my screen and go over the slides with you. What I like to talk to you about is our recent efforts to define what are the units of diversity, of microbial diversity and communities, and also some more recent efforts to track these units over time and over environmental perturbation and understand how this diversity is maintained, essentially. So I'm going to use ANI a lot in this talk, and so I wanted to define ANI first to make sure everybody's on the same page. So ANI is basically a measure of overall genomic relatedness. And the way you do it is you compare two genomes in the old times with BLAST to figure out what genes they have in common, and ANI is simply the average nucleotide identity of the genes they share. So if they share 2,000 genes, what is the nucleotide identity of these genes? In the old times, as I said, we did it with BLAST, but it doesn't scale up, and now we have hundreds of thousands of genomes. So recently, with some computer scientists on Georgia Tech campus, and specifically Chirag again, who is actually now an assistant professor in India, back to his home country, we came up with FastANI, which basically computes very accurately ANI values, but using the k-mers, and it does it 50 times faster or more. And we have shown with Chirag that is a very accurate method, and some alternative implementation, like for example, mass, they don't do as well when we have fragmented genomes or genomes that are contaminated compared to FastANI. So having this tool now, we can go ahead and analyze all genomes in NCBI in a few hours. And that's what another postdoc and former graduate student in the lab, who is now also an assistant professor in University of Pittsburgh, Luis Miguel Rodriguez, did. He used FastANI to calculate the ANI values among the 90,000 genomes that are in NCBI. And what you see here is the key graph of that work, of that paper. And to our surprise, what we saw was that the ANI values among these 90,000 genomes, so we are talking about 8 billion pairwise comparisons. That's what is summarized in this figure. They are either above 96 when they are closely related. And most of these are actually already in the same species. So the way we have been describing species seems to be consistent, with a 95, 96% ANI threshold, or they are distantly related. They are below 85, and there is a lack of data. There is a clear bimodal distribution between 84 and 96. And that's what we call gap in ANI, meaning there's much fewer points there. It's not 0 points, but there are much fewer points compared to either being very closely related or distantly related. And so this was quite surprising to us, because it indicates that maybe species exist, and the 95, 96% ANI is a good threshold to define them. And I want to make a comment here that if you use universal genes, like for example, the MOTU paper that uses a handful of universal genes, about 100, you don't see the same pattern, because these genes are very conserved. And so they are more conserved than the genome average. And when you use the genome-wide ANI, you get a clear picture. This was surprising to many, including myself. And so there have been recently a couple of papers. You see here some citations that question that gap, and they say that it's probably cultivation biases. Now, it's not so easy to think that there are major cultivation biases between organisms that are related at 90% ANI, etc., because these organisms are still very closely related. Like, for example, E. coli versus Salmonella will be somewhere here around 82% ANI, and they can grow in very similar media. And so that idea is a valid one, is a valid concern, but it's hard to see how it will apply when organisms are still very closely related as these organisms are. But to further address that point, we switched to metagenomes, because a natural population, when you study with metagenomes, you shouldn't have any cultivation biases. The approach we took is shown here on this graph. And so the idea is, let's focus on the red population that is in that metagenome sample, wherever the metagenome sample is coming from. The idea is, if there are sequence discrete populations like what we have seen based on the isolated genomes from NCBI that I showed you in the previous slide, then we should see a picture like this. If we lay down the red genome, the representative red genome, and we map all the reads against it to form these read recruitment plots, we should see that the red reads map evenly across the genome and their identities are above 95. And then all the reads that represent the blues, the greens, the yellow populations, they should be below 90% ANI, a nucleotide identity. In this case, it's a single sequence. And there should be a gap, a shortage of reads between 95 and 90%. The conserved genes will be an exception, but overall you should see something like a gap. And if on the other side, it looks very it looks like cultivation biases are playing a big role, we should see something like this, like all reads, all kinds of identities, there is a continuum and it's not clear to separate. To make a long story short, also because this work has been published recently in Nature Communications, we look at many metagenomes, hundreds of metagenomes from the soil, ocean, and we almost always saw this sequence discrete population. So here is an example from a MAG of an unnamed yet uncultivated actinobacterium that we recovered from the 100,000 meters in the Gulf of Mexico, mapping against the metagenome from the same sample. And so what you can see, this is a busy plot, is the new recruitment plots for Illumina data. They have been described recently also, and they are free to use. And they have some very nice panels, like for example, panel 2 shows you the coverage across the genome. But I want you to focus on the main graph, and basically what you see there is that the reads from that metagenome, either they are very identical to the reference Actinobacterium MAG, or they are divergent and there is a clear shortage of reads around 95% nucleotide identity. And to see that even more clearly, focus on this panel number 3 here on the right, where it shows you how many base pairs are there in every unit of identity. So you see at 99 and 100%, we have about 10 to the 5th. And then when we go to the 95, it's dropped to more than an order of magnitude, 2 orders of magnitude actually. And so basically, the picture we see from metagenome is strikingly similar to what we have seen with isolates. And so I'm more convinced now that there are units there, and with these metagenomes, we can track them over time, etc. So I'm going to show you an example of tracking some of these communities from a specific project that I'm doing with a great collaborator of mine, Ramon Roselo Mora in Spain. We are studying these saltans. So the saltans are the environments where we get our salt to eat. And they are very reproducible kind of communities. There is water that is put there. The sun evaporates the water. The salt concentration is built up and eventually precipitates. And that's the salt that is harvested for human consumption. So these communities are very low diversity. They only have 3-4 major clades. 2 are archaea. 1 is bacteria. The bacteria is the Salinibacter ruber. And they have a lot of intra-population diversity, but not very diverse overall. And the other key aspect is only 2 are the major environmental factors. So light for the evaporation of the water and the salt that is built up. So in terms of complexity is not so complex, and that's a big advantage because you can track them, etc. So with Ramon and help actually from JGI, we have sequenced 123 from one of the CSP programs. We have sequenced 123 Salinibacter ruber from this point in Mallorca, in the middle of the Mediterranean Sea. Very nice environment, a place to be and work. And what we are trying to understand is what is the diversity of these genomes, how it is affecting the population, the function of the population as the conditions change, salinity and light primarily, and so on. So with the help of 2 great graduate students, Roth Conrad at Georgia Tech and Tomeu Viver in Mallorca. Tomeu is now a postdoc at Max Planck. We analyzed these genomes. What we found was it has a very interesting structure. So here what you see is a comparison among these 123 genomes, and it shows you the ANI on the x-axis plotted versus how many genes they share from the genome. And so what we saw was that there are 2 dominant populations, an abundant one and the less abundant one, and there is a clear ANI gap between them. In this case, the less abundant to the most abundant is 95% ANI. And then the genomes that make the most abundant population, they are 97 to 98% ANI among them. So we focus on the most abundant population. So even this is an interesting case to study the sequence discrete population and how they are evolving. For the rest of the talk, I'm focusing on the most abundant ones. We did a pangenome of them. So here is how many genes they have unique among the 100 genomes, like subsample to 100 genomes. And to our surprise, if you like, we found that it has 40,000 non-redundant genes. So quite big as a pangenome for 100 genomes. And actually, it's about the same size as E. coli. If you take 100 genomes from E. coli, from NCBI, you will see a very similar pangenome, but the E. coli genomes come from many studies, many samples. Here we are talking about the same location, adjacent ponds that are communicate, the water communicates between them. And so this is a picture from Roth's papers to show you that basically the genes among these 100 Salinibacter or 123, they are either core, like everybody has, or strain specific. There are very few genes that 20 or 30% or 50% of the strains have. And then if you look at the strain specific genes, if you see here the alpha, the A is the Salinibacter, B is the E. coli, and it has a lot of other species. The E. coli and the Salinibacter are very similar, and Salinibacter appears to be even more diverse than E. coli, like having more unique genes, more strain specific genes than E. coli. And so this is quite surprising to us because there is a lot of diversity on these environments for the bacterial fraction. And so one big question was, what is the value of this diversity when conditions change? So with Ramon, what we have done two key experiments manipulating the two key variables of the site. So you can see here the schematic. So basically we took a pond that is saturated for salt, so salt starts to precipitate, and we shaded it with a mesh on the top. So very little light was penetrating through. Excuse me. And then what we did, we also dilute another pond that it was also close to saturation with a tap water. So we drop the concentration of salt from 34%, which is the saturation conditions, to 12%. And then we follow this pond as well, the control that was ambient light and no shading and etc. We follow it for 1 month, the first day, first week, and 1 month, when basically the conditions have resumed to be saturation conditions again. And we took metagenomes and we wanted to see from all these strain-specific genes what are important, etc. So this is a key figure from the paper. So what we saw was under control, nothing changed. The abundance of the genes and the strains were the same. The same appears to be for the pond we shaded, that there was very little, change. However, the one that was diluted and dropped the salinity to 12%, we saw that 1 week later, when salinity is intermediate, there were a lot of genes, several genes that were spiking at abundance. Actually, they are not a lot. It's about 2% of the total pangenome, so not a big fraction. And then when we annotated, we saw that they were all related to osmoregulation, sensing the environment. So we believe that these genes are ecologically important and gave an advantage to the organism that carries them. However, 1 month later, when conditions changed, these genes went back. And actually, if we look at the isolates we got 1 month later, they are very similar to the ones before. So even though they are ecologically important, they don't seem to purge the diversity. They don't seem that the organisms that have them can outcompete the rest. And 1 month later, you see all the diversity coming up. And so this was a little surprising also because we thought that they will, reduce the diversity and those that have the ecologically important genes will dominate. So our working hypothesis right now is that they are ecologically important, but the advantage is not big to purge the diversity. And it's only small, especially with respect to the generation time, which is 1 generation per 2 days, so they grow not very fast. And we're currently testing that hypothesis using some lab-specific competition experiments. But it does look that, the diversity is maintained, even though there are ecologically important genes, and maybe the key here is how strong the ecological advantage is. I'm going to finish with some unpublished work coming out from this project, which I think is also very exciting. So in our analysis of these 123 genomes we got, we saw another gap of ANIs. So more specifically, we see a shortage of genomes, even though we randomly sequence them with no pre-selection, etc., around 99.5% ANI. So either our genomes are very, very similar, or they are below 99.5. And so that gave us the clue then to look at all our genomes more broadly. So we went back to the NCBI genomes. We found 300 bacterial species that they have at least 10 representatives. And we do a very similar analysis. So using 300 organisms now, and let me zoom in that specific area of interest, the 99 to 100%, we see another kind of gap. And you see, there's very few points there. There are points, but a few, and the genomes are either more than 99.9 or below 99.4, etc. So we think there is another interesting ANI gap, and that may be used to catalog the diversity within the species now. And so we overlay so here we are looking at the E. coli data only, and looking how the sequence types are comparing to that 99.5% ANI cutoff. And so sequence types are defined by having identical or almost identical alleles in 5 or 6 loci. These are usually universal genes, highly conserved genes. And so what you see here is, for example, the ST11 of E. coli is very consistent with that idea, meaning they're either part of that ST, they're very close related, and they don't have any other relatives. The other relatives are below 99%. And here is a case where you see that this ST, the ST10, is probably too broad, but it does have that gap around 99.5. And then there is also the ST165 that is very closely related and overlapping. And so what we are saying is we can use this 99.5% to call the STs more accurately and also make it automatic so you don't have to manually curate, etc. And then for us, our proposal would be that the strains will be at 99.99% nucleotide identity. So strains will be, just to account for the sequencing error and so on. And for the sequence types, we propose the 99.5 as a more reliable and automatic threshold and so on. And there's very similar data for other groups as well. So to summarize, what I hope I have shown you is that there seems to be that prokaryotic species exist, and that 95, 96% ANI is a good threshold to define them. Not always, but in 99% of the cases. There's a lot of 90 plus in this talk. The metagenome picture is strikingly similar to the isolate picture and help us identify these organisms and track them over time. When we did that for the Salinibacters that dwell, they like it in the Saltans, we saw that the majority of the pangenome is not changing. It seems to be ephemeral and not ecologically important, but a few of the genes seems to be ecologically important, but maybe the advantage that they confer is not so big to purge the diversity. And we have also observed a new kind of gap within the species that can help us define sequence types and based on sequence type clones, clonal complexes, and also strains. If you have any questions about the talk or you want to join the group, please shoot me an email at kostas@seed.gatech.edu. And I want to say that the majority of this work is supported by NSF, but also the community sequence program of JGI have sequenced a lot of our strains. Thank you very much for your attention.